Right, well this is the final video in the protein synthesis topic 14, and it's just the end bit of the notes, which is chromosome mutation. But it does give us an opportunity to recap meiosis and your meiosis knowledge, because this really is an example of mutations that result in incomplete meiosis or events of meiosis. So let's get on to this one. Okay, so this one is the final PowerPoint, PowerPoint 4. Okay, so this is called chromosome mutations, and it links to your keynotes, just a tiny section of your keynotes, just one page. Okay, so chromosome mutations, these are mutations in the number of chromosomes, and they're spontaneous events as well, but these occur during meiosis. So when we're producing daughter cells by meiosis, if the division stage doesn't go as it should, okay, then you can end up with not separating the homologous chromosomes or the sister chromatids during the division stages, and that results in having more chromosomes in gametes than you should have. Okay, so we'll come back to these keynotes once we've done a little bit of key work reviewing meiosis. Okay, and there's a little bit at the beginning of this PowerPoint as well where we can just look back over things we did last week, just test our knowledge. So can you have some pen and paper at the ready, and we're going to do a little quiz to start just on some of the key knowledge from last week. Okay, so can you write down on the paper in front of you the definitions of proteome and genome? Okay, have you got it down? So we should have, for proteome, all the proteins a cell can make, and for genome, the complete set of genes in a cell. There's a different way of saying that as well. So if you've got all the genes of a cell, that's fine as well. All the genes of an organism is fine as well. Okay, what about this table? Can you please pause while you copy it down and complete it? Okay, you should have completed now. So hydrogen bonds, present or absent, and number of polypeptide chains. Well, DNA does have hydrogen bonds, it's 2 polypeptide chains. mRNA doesn't have hydrogen bonding, although bases are exposed, but it is a single strand, whereas tRNA is that single strand clover-shaped molecule, so it's folded back on itself, and that required hydrogen bonds to hold that shape. Super. Right, what are mutagenic agents? Can you give me some examples? You'll need to pause while you write this down. It'll take you a minute or so. Okay, are you ready to review? Mutagenic agents increase the rate of mutation, which of course are changes in the base sequence of DNA during DNA replication. So they're anything that increase the rate of that happening. What about UV light? What about ionizing radiations, like gamma radiation, or a named chemical like benzene? Okay, so thinking about what we did last video now, here we've got 2 amino acids coded for, okay, and we're going to delete the sixth base G in the sequence shown in the diagram, which we haven't got. Ah, there it is. Okay, so here is the base sequence on the top. So deletion of the sixth base, 1, 2, 3, 4, 5, 6. So deletion of this base here, so RG, in the diagram above, would change the nature of the protein produced, but substitution of the same base would not. Okay, you're going to have to write your answer down for this. Use the information in the table and your knowledge to explain why. So if we were to delete the G, we'd get a frame shift to the left. The U would move to here, the A to here, etc. What would that mean in terms of the coding? Okay, so what amino acids would change? Because they've told us it would. But then our substitution, so if we change that G for an A, if we change it for a C or a U, what would change? Would anything change? Why? Okay, so have a go. Okay, so have you got your answer ready? Okay, so if we did take that G and we deleted it, okay, that would mean that this would become C, C, U. Okay, so it would change from being C, C, G to CCU. It wouldn't have an impact here, but it would mean that the next codon would be A, CC instead of UAC, and so on. So you change every one. Okay, but if you just substituted that G for any other base, it would still code for proline. Okay, so substitution would result in that one, and that one, they all code for proline. Deletion would cause a frame shift, that's to the left, and that would change all the following codons. So for example, the next one. Okay, right, back on it then. Oh, no. Nearly gave the game away then, sorry. Okay, so what is the role of the of RNA polymerase in transcription? Write it down. You'll need a minute or so to do it, so pause the video while you do. Okay, so are you ready for your answer? Okay, its job is to join adjacent RNA nucleotides together by condensation reactions, forming phosphodiester bonds between the sugar and the phosphate. And so produce a mRNA copy of the template strand. It also moves along that template strand from 3 end to 5 end. That's just a side note. Okay, following transcription, the RNA in the eukaryotic cell is shorter than the gene used as a template, but in a prokaryotic cell, the mRNA is the same length as the gene used to produce it. Explain this difference. Again, pause and write down your answer. Okay, you ready for your answer? So, DNA in a eukaryotic cell has introns within the gene, prokaryotes don't. So after transcription, these regions are removed from the pre-mRNA in a eukaryotic cell, okay, by the process splicing, I would add. Okay, next one. Can you put these events in the correct order? So you're going to have to pause and give yourself a little bit of time to write the letters down in the correct order, starting with the earliest. Okay, so you should be ready. Joining me back. So it was the strands separate first, then the RNA nucleotides join to expose bases. Okay, then we skipped quite a lot there because then the RNA polymerase would join them together and produce the mRNA. Then the mRNA leaves the nucleus, attaches to the ribosome. The tRNA brings the specific amino acids, and finally peptide bonds between the amino acids. Okay, right. So that's the little intro quiz complete. That's recap some of your knowledge, but not the knowledge we need for this last little bit, this last section of this topic. To be able to do that, you need to have a good understanding of meiosis. So this is found in your topic notes for topic 8. Okay, if you do need to have a good look through and a read of that before you commence this next bit, I am going to do a bit of a recap for you. Okay, so meiosis, of course, is the type of cell division which halves the chromosome number. It does this by dividing this, or the cell does divide twice. First, in the first division, the homologous chromosomes align on the equator and are separated into the 2 daughter cells. So each daughter cell has got 1 of each chromosome pair. So at that point, we've halved the chromosome number. But we still have 2 copies of each of those chromosomes within each daughter cell. So they proceed to divide a second time. This time, when they line up on the equator, the sister chromatids are separated, and then we have our 4 daughter cells. Okay, in terms of a bit more detail, so if you think about that first division, there were some events that happened in the first division which are incredibly important. So this is our cell that's about to divide by meiosis. Okay, you can see it has 4 chromosomes and they're arranged in their 2 homologous chromosome pairs. Okay, so we'll call this pair chromosome 1 and we'll call this pair chromosome 2. Okay, so this person can't be a person, or 4 chromosomes. This organism has 1 chromosome 1 that it's inherited from its father, we'll say that's the blue one, and 1 that is inherited from the mother, which is the purple one. Okay, and the same for chromosome 2. But because DNA replication has occurred just before this cell division started, when the chromosomes condense in prophase 1, in the first part of the first division, they are chromosomes containing 2 identical copies. So each DNA molecule has been copied and then condensed and held together in 2 sister chromatids held by a centromere. Okay, so the first interesting event of meiosis does happen right at the start here, where the homologous chromosomes have got together, they form bivalents, so they kind of join together in the homologous chromosome pair, and this process takes place. It's called crossing over. The homologous chromosomes become really closely associated with each other in something called a bivalent, and then they exchange the tips of what we can refer to as the non-sister chromatids, because they're chromatids from either homologous chromosome, and they exchange tips at the Chesmata points, the crossing over points. That means now that each one of these DNA molecules are slightly different to each other. So this process was to increase variation by mixing alleles. Okay. Once that's over, the homologous chromosomes align on the equator. Now, they don't do this in single file in meiosis. They do this in the homologous chromosome pairs. So they sit next to each other on the equator with a spindle attached to their centromere. Which side they take of the equator is random. So we call this process independent segregation, because we're going to pull the homologous chromosomes apart, but which side, which daughter cell they end up in, so which side they're pulled to, depends on which side of the equator they are sat, and that process is independent. It's a random process. Okay, so following the anaphase 1, each daughter cell will contain only 1 of each homologous chromosome pair. Just to show you all the different ways a cell with 6 chromosomes could arrange its 6 chromosomes in that process. And that starts to show you the type of variety that can be produced by independent segregation as well. Okay, so both crossing over and independent segregation introduce genetic variation through the mixing of alleles during meiosis. Okay, so then in anaphase 1 we can see our homologous chromosomes have been pulled to opposite cells, so when they divide we've halved the chromosome number. Okay, so the 2 daughter cells that have been produced now are haploid. They only contain 1 of each homologous chromosome pair. Also note the sister chromatids are not truly identical anymore because of that crossing over with the non-sister chromatids. So they're in effect different DNA molecules, these. Okay, so the second division is all about now, well, we've got 2 copies of each DNA molecule. We don't need that, so we're going to halve the DNA content. Okay, so this looks more like mitosis now. The chromosomes align on the equator and the sister chromatids are pulled apart this time. Okay, and that means that now we've got 4 daughter cells that are produced during meiosis, and crossing over and independent segregation or random assortment has taken place, and that means that the genetic combinations of alleles in each of those daughter cells is different to each other. Okay, if you feel like you want to recap your meiosis further, then what I have included in the PowerPoint is a link to an animation for you. So I'm not going to show you that, but if you want to, just have a little moment to compare mitosis and meiosis again. Make sure you've got really good, strong meiosis knowledge before you move on to the next section. Please do. Okay, so assuming now we remember our meiosis, let's have a look at an exam question where you are describing the process of meiosis. So describe what happens to chromosomes in meiosis. So this is what a good one looks like. This is a waggle. Okay, so what would you do? How would you describe this, and what would your answer look like? And then read this one and tell me what went well. Okay, so read this 3 together. The chromosomes condense and arrange into the homologous chromosome pairs. Crossing over can then occur between the tips of sister chromatids. Now you might be better calling them non-sister chromatids because they're the chromatids of separate homologous chromosomes. The spindle fibres attach to the centromere and move the homologous pairs to the equator of the spindle, where they are side by side in their pairs. The side they take is random, and this process is called independent segregation. The homologous chromosomes are separated to opposite poles by the contraction of the spindle fibres. This is the first division, and it halves the chromosome number. The daughter cells divide again. This time, chromosomes are separated into their sister chromatids. Again, spindle fibres attach to the centromere and contract, fracturing the centromere and separating the sister chromatids to opposite poles. Okay, so where were their mark points? Now they could have put them in various places, and in fact they could have asked for some detail this student's forgotten, which might have let them down a little bit. They actually hit the mark points pretty well for this SMART scheme. So there was a mark point for describing crossing over. So you didn't get a mark point for crossing over on its own. You had to have the whole detail. Okay, what I think now on the new specification you'd have to add is the chasmata points, and be very careful to say that they're non-sister chromatids that are exchanging tips. They also got a mark point for independent segregation, okay, describing that process. They got a mark point for saying that homologous chromosomes were separated in the first division, and that therefore halved the chromosome number. And they got a mark point for the detail of how the spindle fibres attached, and that the sister chromatids were pulled apart in the second division. Okay, so have a go at this question. Meiosis results in genetic variation in the gametes, which leads to variation in offspring formed by sexual reproduction. Describe how meiosis causes this variation and explain the advantage of the variation to species. Okay, so you've done this question before. I think it's in the question pack for meiosis. I want you to have a go at it again now from memory. Okay, so would you like to now compare your answer to the mark scheme? Okay, so the first part was, where did this variation come from? So we want crossing over. So at the beginning of the first division, the homologous chromosomes align, and at the chiasmata crossing over points, non-sister chromatids are exchanged within homologous chromosome pairs. Yeah, so something detailed. So you don't just get a mark point for crossing over, you have to describe it. Again, independent segregation described. So within the first division, so within meiosis 1, the homologous chromosomes align randomly on the equator in their pairs. Which side of the equator is random for each pair. Okay, and so when they are separated, there's that random segregation, as we showed in the recap with this chromosome, the cell with 6 chromosomes, how many different variants you could end up with that random segregation. And a third mark point. Now, I don't know if you remember this one, but because at the point of these chromosomes lining up in the second division, they're no longer identical DNA molecules. So you've got a second independent segregation, because that chromosome could have been facing the other way. And so you've still got a randomness to which single DNA molecule ends up, which combination of those single DNA molecules ends up in the daughter cells. So there's another mark point on this mark scheme saying you've also got some independent segregation of the chromatids in meiosis 2 in the second division. So that is further adding to the variation. Now, you didn't have to have that, to be fair, but it's an extra mark point, and we always want an extra mark point. We never want to not give them the detail. Okay, so then it was answering the second part of the question. Explain the advantage of variation to the species. Now, we haven't done this in any great detail at A level, but you can see these aren't really very difficult mark points. So it's that idea that variation in the offspring means we've got individuals who will be better adapted. They will survive, reproduce, and pass on the alleles and genes, the advantageous ones. This allows for organisms to survive changes in the environment, or a change in competition, or whatever it is you decided to describe. Okay, so one last, and this one's quite a tricky one, recap question on meiosis. Okay, so for this you will need to pause because you've got to do some drawing. Okay, so you need to draw this diagram, or you can print it out from the PowerPoint if you like. We've got a cell with 6 chromosomes, okay, 3 homologous chromosome pairs, and I've paired them up, you can see, in terms of the size, because the homologous chromosomes are the same size. Okay, so now what I would like you to do is to populate the diagrams to show the first division, so how would they look during the first division, anaphase 1, at the end of the first division, and then through the second division and the end of the second division. So show me what the chromosomes would look like in each of those cells. Okay, and then join me back. I'm going to show the answer now. Okay, so in the first division, it's weird, but in anaphase 1, you kind of have to draw the homologous chromosomes being pulled apart. And so they form this kind of V shape, but because you've got like a whole chromosome, it's kind of got 4 pointy ends, if you like, coming back in a V shape. So to show them like that would be perfect. The result then is 2 daughter cells, each with 1 of each homologous chromosome pair in them. Still during prophase the same. In metaphase 2, then they're all lined up in single file on the equator. And then at the end, obviously we've separated the sister chromatids, so each cell would have now just 1 copy of each of the homologous chromosomes, because we've halved the chromosome number. It's a haploid cell. It's only got 1 of each homologous chromosome pair, and it's only got 1 copy now of each homologous chromosome. Wow. So if you've managed that, you really know your meiosis. If you made a few mistakes, don't worry. Okay, it's tricky, this question, but what you do now need to do is make sure you understand that process. Because we're going to move on to chromosome mutation. And chromosome mutation is basically when this goes wrong. And where it goes wrong is the separation steps, either in metaphase 1 or metaphase 2, and anaphase 1 and anaphase 2. So when the homologous chromosomes or the chromosomes line up on the equator and how they separate, it's when that process goes wrong is what we're going to look at next. Okay, so before we do, can I ask you to go to question 7, just to make sure our meiosis knowledge is practiced, and complete that question in the question pack, and then come back. Okay, so when you have completed question 7, I'm going to run through a good example of the answer to question 7 in the PowerPoint. Okay, so I'm moving on to that now. Okay, so you were asked to complete the table by ticking the cell that shows the feature. So B, C and D, the first thing I did was count my chromosomes and look at what was happening. So they told me they were all from the same organism. So I know this is a haploid cell and that these chromosomes are on the equator. So I know this is in the second division because the homologous chromosomes have already been separated. Their number has been halved. On this one here, I can see this is before the homologous chromosomes have been separated and something's going on between them there. They seem to be crossing over. Okay. And then in this picture here, this is looking like mitosis to me because the homologous chromosomes have lined up on the equator, but they're not lining up in pairs, and the sister chromatids are being separated. So I've identified that, and I probably would write notes around, and from that I've been able to populate, or the student's been able to populate the correct answers in the table. So homologous chromosomes are present, well, they're present in B and C. And of these, which one was in mitosis? That one, because this one was stage 1, the first division of meiosis, and this was the second division of meiosis. Evidence: homologous chromosomes in pair, that only happens in meiosis, and we've halved the chromosome number, so it must be during meiosis. Okay, describe and explain the appearance of chromosome K in cell C. So describe. Each chromosome is made up of 2 sister chromatids joined by a centromere. So that's the description. Tick. Why? So I've got to explain why. Well, each sister chromatid is a copy of the same DNA molecule produced during DNA replication, so that each daughter cell gets a copy of each DNA molecule. So that's why it appears that way. So my second tick is the for the why, explaining why it appears that way. Explain what is happening at point J in cell B. So if we go back to cell B, and this is point J there, we can see crossing overs occurring. It's just making sure we get the detail in there. Okay, so in meiosis, first metaphase, the homologous chromosomes line up on the equator. At J, crossing over is occurring. Now we know that doesn't actually happen in metaphase, but the examiner's using what they can, which is, you know, if they show you crossing over, you say that's what it is. Okay, so the mark point is actually here, and it's for the detail. So at J, crossing over is occurring between each of the sister chromatids of one homologous chromosome pair. Now I said it was actually better to describe them as non-sister chromatids because it's less of a mouthful to write that. So if you put of non-sister chromatids, that means the same as each of the sister chromatids of one homologous chromosome pair, which is a bigger mouthful. And then a mark point then for the detail. At the chiasmata, the sister chromatids swap sections, exchanging alleles between non-sister chromatids. Okay, so at the chiasmata points, that's where the non-sister chromatids exchange alleles, swap sections of chromatid. Okay. Part D. Use the information in all three cells to explain the number of chromosomes in cell D. Okay, so cell D has 2 chromosomes, so we've had a halving of the chromosome number. So the organism has 4 chromosomes in a normal body cell, so it's diploid, but during meiosis, the first division, the homologous chromosomes are separated into each of the 2 daughter cells, halving the chromosome number, which is explaining what's in that diagram. And then finally, explain why some cells contain a mass of DNA between 1 and 2 arbitrary units. Now, this really gets some students. And really, you just got to think about, well, when you've got 1 that's before DNA replication, 2 is after DNA replication. So what about if some cells are in the middle of it? So that would explain why you've got some mass in between, depending how far through DNA replication they are. Easy when we know the answer. Okay, so you can have a look in your keynotes alongside this if you like. I'm going to talk through now chromosome mutation. So again, we're going to come back to chromosome mutation in year 2. We're just going to look at some simple chromosome mutations caused by non-disjunction during gamete formation first. Okay, so if you look at these 2 pictures, you can see there are 2 places that meiosis is going wrong. Okay, so in this on the left hand side, you can see during the first division something has gone wrong, and it's this process here where the homologous chromosomes are usually separated so that one of each pair goes into the daughter cells. You can see that that's not happened here. We've got non-disjunction, so both homologous chromosomes have ended up in this daughter cell, which means that when the second division is over and the sister chromatids here have been pulled apart, we've got some gametes with no copies of one particular homologous chromosome, and then other gametes that have got 2 copies of that homologous chromosome. So this is an example of non-disjuncture, and that's causing a mutation, a chromosome mutation. On the other side of the picture, you can see non-disjunction at a different place. So it's not in the first division, but it was in the second division. But it still resulted in cells that have got more information. In this case, sister chromatids didn't separate, and one daughter cell received 2 copies of the same DNA molecule, whereas the other one, other daughter cell in that line didn't receive that homologous, any copy at all of that homologous chromosome. Okay, now these chromosome mutations mean that if these gametes were to fuse with normal gametes during reproduction, sometimes we're missing chromosomes, and in other cases we have got more of a particular chromosome than is required. Okay, so the most common chromosome mutation, I think the one that you'll probably know about already, is Down syndrome. So Down syndrome is caused by having an additional copy of chromosome 21. Okay, so about 1 in 1,000 babies in the UK, the rate is 1 in 1,000 babies in the UK are born with Down syndrome. So it is quite a common chromosome disorder, and it does affect people in a variety of ways. Particularly, it can cause heart problems. So about half people with Down, or half children with Down syndrome, need some sort of cardiac surgery for heart problems associated, and it can also lead to learning difficulties as well. Okay, so that's probably the one you know about. Other ones that you've probably not heard of. Okay, Turner's syndrome is another example of a chromosome mutation, and this is where girls are born with only one X chromosome instead of two. And some of the characteristics that causes are shortness of stature and a sort of webbing of the neck. Okay, so there are a couple of characteristics. So one celebrity, Linda Hunt, who has Turner syndrome, quite a famous actress you might know of. One other example is Klinefelter syndrome, and this is where males are born with an additional X chromosome. Okay, so they have an additional X chromosome, and so although male have excessive breast tissue, some, and other things associated with the syndrome, being rather tall, quite youthful looking as an adult. Okay, so as a bit of a challenge, I found this diagram online because we haven't had any really good exam questions yet on non-disjunction mutation. I've got one example I'm holding back for a homework, but this is a similar sort of thing to what they could ask. Okay, so there's two diagrams to illustrate how someone might inherit Klinefelter syndrome, so this additional X chromosome. In the pictures, you can see, if we talk you through A, that in the first division, something's, and then the second division. Okay, what happens in terms of gametes, and then the same over here. You can see that we've got an issue in the production of the gametes here. Okay, so have a go at trying to work out what's gone wrong, and then join me back. Okay, so A shows, it's a bit tricky on A, shows non-disjunction of the homologous chromosomes during division 1 of meiosis, so that homologous chromosomes are not fully separated. B shows non-disjuncture of the sister chromatids. That one's a bit easier to spot. But what both show is that the gametes that are produced, that are the faulty ones, are the ones that contain additional chromosomes. So this is from mother and this is from father. So in this case, Mother has produced a gamete with 2 X chromosomes in. When it fuses with the gamete containing a Y chromosome from the male, you end up with 2 X's and a Y. In this example here, we've got a gamete produced with an X and a Y from the male, which when it fuses with a normal X containing gamete from the female ends up with XXY as well. So in both cases you've got 2 copies of, of a chromosome, which when fuses with a normal gamete results in 3 copies. In this case, XXY. Yeah, so sometimes it's 2 X's from the female, sometimes it's an X and a Y from the male and an X from the female. We've got 2 copies of a chromosome, and the normal gametes 1 copy, resulting in 3 copies, in that case XXY. It's interesting. Okay, so that's one type of chromosome mutation, and it's the type that's caused through non-disjunction. So let's have a look back in your keynotes. Okay, so in your keynotes you can follow alongside there. Mutations in the number of chromosomes can arise spontaneously by chromosome non-disjuncture during meiosis. So meiosis produces daughter cells that are genetically different from each other. So this is just a recap of meiosis. Remember, there are 2 nuclear divisions where you would usually form 4 haploid daughter cells from a single diploid parent cell. Remember that in normal meiosis, genetically different daughter cells have resulted from independent segregation of the homologous chromosomes, where 2 to the n gives the possible number of combinations. If you think back to that chromosome picture, you can check it out again. There were 6 chromosomes, so 2 to the 6. And if we pop back into the PowerPoint, we had 8 shown. I don't know if that's all the possible combinations. You can check it out. And that's without even considering crossing over. Remember, crossing over is another place where non-sister chromatids swap regions of DNA at their chasmata points. Equally, on top of all of that, we've got random fertilization of the haploid gametes increases genetic variation. So that's the normal meiosis recap. Okay, but within meiosis, the cell division of each daughter cell contains 1 of each homologous chromosomes. Sometimes it goes wrong and daughter cells are produced containing too many chromosomes. Okay, it may be a pair of chromosomes fails to separate in the first metaphase, or in the first anaphase really, or sister chromatids fail to separate in the second division. Okay, and you've got that lovely diagram. You do need to know just this one example, which is the Down syndrome example, non-disjunction of chromosome 21. So you end up with 3 chromosome 21s. Right, whiz back to there. Okay, so one thing to look forward to then, mutations come up again next year. So we'll be doing mutations at gene level. So that's when we would be doing addition as well as substitution, inversion, etc. But we're also doing them at chromosome level again next year. So we'll be looking in more detail. So if you do Google chromosome mutation, you'll find diagrams like this showing all this different variety of chromosome mutations that happen, as well as non-disjunction ones that we've looked at this year. Okay, so that is the end of the topic. Okay, there are still a couple of questions in the question pack to finish off. So I'd like you to complete your student note revision questions now and check their answers in the mark schema on the Quizlet, which I have shared with you. Those people are in the Quizlet group. If not, I will put the link in the log for this week's work. I've added some extra practice Quizlets, including some fast track sets. The fast track sets are really about linking topics and developing ideas a little bit further now, going into year 2. I've also put in there a full year revision set, which is something that you might sort of pick into and out of as you're keeping all your knowledge nice and keen, ready for developing it on a little bit further. Complete the question pack, check it, mark those remaining questions. There's a spot test left to do, and remember in the first PowerPoint there was an extension task which you can complete, and there is one in this PowerPoint as well, so I'll just remind you of the extension in